right, so Chapter 12 covers the central nervous system, and that is going to be the brain and the spinal cord. So what we're talking about here in terms of the overall nervous system, we divided into the central nervous system and the peripheral nervous system. We saw that in Chapter 11, but this is a pretty artificial divide in the sense of one cannot function without the other. Okay, you really can't understand what the central nervous system does without having some idea of the peripheral nervous system, and vice versa. Right? But we're going to try to focus in on the brain and spinal cord in today's lecture. Now, as we go through this, we have to start off with what's called cephalization. Right? We are going to talk about the brain, specifically looking at the human brain. Right? Any vertebrate mammal, though, so whether we're talking about humans or dogs or sheep or whales, right, have quite similar brains. Not exactly the same, but similar. So in lab, we will actually look at a sheep brain and kind of take apart a sheep brain rather than looking at a human brain. All of these vertebrate mammals, humans being included in that, have a high degree of cephalization. Right? What cephalization is, if you see cephal in the beginning, you might think back to that very first lecture that we did with the anatomical regions, and remember that cephalic means head. So cephalization is where you have an elaboration of the anterior portion of the central nervous system. Okay? So we have a focus of the central nervous system in the anterior or front portion. Now in humans, anterior is superior. Okay? So those words kind of flip in humans. You could imagine if we were a goat or a sheep, our brain would be anterior. Only humans have it superior. What we're going to see is that we're going to have a huge increase in the number of neurons in the head. We're going to have a focus of neurons in the head. Our special senses are going to be located in the head. Your book talks about how the highest level is reached in the human brain. We can argue that a little bit. There are certain things, obviously language, communication areas, memory, processing, those kind of things are highest in the human brain. But there are some vertebrate mammals that have other parts of the brain that are much more developed than in humans. We have really crappy sound, really crappy hearing, I should say. We don't pick up sound very well. There are many mammals that hear much better than we do. The highest level of hearing, the hearing portion of the brain, is found in a mammal. Does anybody want to take a guess of what mammal has the most complex hearing centers? What'd you say? Not dogs. Dolphins. Dolphins. Dolphins can echolocate. They basically can use their own sounds to create visual patterns. Well, not visual patterns. Like echolocation or sonograms that they can interpret. So they can actually see what they're hearing. Right? Pretty cool stuff. We can't do that. Right? If we were to shut off all the lights and put you in a very, very dark room, you could not go beep, 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 and know where you're going. Right? It'd be awesome if you could. It'd be awesome if you could. But we have a highly, highly advanced brain and lots of neurons in our brain. Most of our central nervous system is located right here in our head. Now we do want to look a little bit at embryonic development. We are not taking an embryology class. If we were doing an embryology class, we'd spend 2 weeks on this. Okay, so we're just going to do an overview of this. The majority of the embryonic development of the nervous system begins very early in embryonic development in the first 26 days. 26 days is like the first month. In a quick nutshell, I've said this before, when we talk about embryonic development, you have a sperm and an egg come together, they form a new cell, that cell starts dividing. That dividing cell eventually becomes a solid ball of cells, okay? That's called a blastula. It's then going to hollow out and fold in, and at that point then we're going to start seeing differentiation. So different cells are going to go down different pathways. So we're talking about the cells that are going to become the nervous system. Where they come from is part of the ectoderm, which is the outer of those layers, is going to fold in, and it's going to create something called the neural plate. Okay? This neural plate is going to fold in, it's going to form a neural groove, which will eventually become a neural tube. So you can imagine if we look at this in cross-section, this part right here called the neural plate is going to fold in, it's going to pinch off, and it's going to become the neural tube. That neural tube is formed by about day 26 of embryonic development. Okay? Okay. That neural tube is going to become the brain and spinal cord. These green parts here that are called the neural crest, those neural crests, those are going to become the peripheral nervous system. What that means is the precursor tissue for an individual is formed by day 26 post-conception. Right? Most human females do not even know they are pregnant by day 26 because it's usually less than one missed period. Okay. So this is one of these things that this might be the most important stuff to form, and it forms before most people even know that they're pregnant, which I think is pretty interesting. Okay. It also does have a lot of consequences in terms of health, and why women who are of childbearing age and are considering having a child are supposed to kind of modify their behavior before they ever become pregnant, because they want to have that, I don't know, a clean slate to start with. So what we're going to do is we're going to take a look at this neural tube. Right? Now you can imagine this neural tube is going to run the length of the developing embryo. This is what the embryo looks like at day 26. Doesn't exactly look like what we would recognize being an embryo yet, but that's it. Okay. This neural tube is hollow on the inside, but it's not empty. It has fluid inside. Eventually it will have fluid called cerebrospinal fluid in it. The anterior region, which will eventually become the head, is also called the rostral region, whereas the posterior, which will eventually become the tail, right, so the back end of it, is called the caudal region. As development occurs, the first thing we see is the tissue, which is going to be shown here as this kind of yellowish, tannish color. The blue is the open space full of fluid. This will start to grow, and we're going to start seeing these spaces. Okay? These are called the primary brain vesicles. So very early on in development, we see 3 divisions occur. We have the forebrain, the midbrain, and the hindbrain. And these are called the prosencephalon, the mesencephalon, and the rhombencephalon. Now, as big and scary as that word is, if you look at encephalon, right here you say cepha, right? Remember we said cephalic means head. We have the prosencephalon, mesencephalon, rhombencephalon. So if you could remember the prefixes and you could remember the encephalon part, it's actually not that hard. These 3 primary brain vesicles, as development occurs, will, by about week 5 of embryonic development, start to form the secondary brain vesicles. Right? These secondary brain vesicles are further differentiation of that initial neural tube. The region that was initially the forebrain or the prosencephalon is going to divide into 2 sections. One is called the telencephalon, the other is called the diencephalon. Now for the lecture, I have broken this down by sections, but in your book and in your lab book, you'll see that this is a continuum in terms of the diagram. Okay. These 2 brain vesicles, the secondary brain vesicles of the telencephalon and diencephalon, we'll spend a lot of time talking about what they do in the adult brain structures. The mesencephalon, or the midbrain in humans, stays the midbrain. It doesn't really undergo any further division. And we will see in terms of adult brain structures, it actually doesn't change very much. It's very limited in humans. Then in the rhombencephalon, or the hindbrain, we will see divisions. In the rhombencephalon, we will see divisions into what's called the metencephalon and the myelencephalon. And if you notice these colors, how these colors change, this is color coding that stays throughout the notes, so you can track where these secondary brain vesicles go or how they develop. So we have the prosencephalon or the forebrain becomes a telencephalon and diencephalon. The mesencephalon stays the mesencephalon. And the rhombencephalon becomes the metencephalon and the myelencephalon. Now this is about week 5 of embryonic development. Here we have the brain laid out in a linear fashion. Okay. Now in some animals, fish for example, the brain stays in a linear fashion. So when you look at a fish brain, you have a front region called the telencephalon, and then behind it you have a diencephalon, and then behind it you have a metencephalon. In humans, though, our brain does not look anything like this. Okay. When we have our change to adult brain structures, we're going to see there's a lot of reorganization in terms of how things grow and the shapes they have. Now, the adult brain structures that come from these secondary brain vesicles are what we will be talking about. So the telencephalon is going to become the cerebrum. The cerebrum is going to be where we will talk about most of our conscious behavior, conscious control of behavior. We will talk about the cerebral hemispheres, the cerebral cortex, the white matter, the basal nuclei. These are all parts of the cerebrum. The cerebrum is going to be the biggest part of the human brain. Right? And it's where we're going to really talk the most about function. Now, the diencephalon will become three main things that are really all together: the thalamus, the hypothalamus, and the epithalamus. And all three of these are considered part of the diencephalon. And also we're going to see the retina. So the retina, which is the part of your eye that actually has neurons and actually receives visual input, that's really an outpocketing of your brain. So individuals who are born with severe brain problems, things that are microcephaly. I don't know if anyone's ever heard of microcephaly. Sometimes babies are born with microcephaly. It's a highly undeveloped brain or underdeveloped brain. They often do not develop eyes, so they often have no eyes. Or they have severe vision problems because their eyes are not normally formed. Okay. The other thing then is the mesencephalon. This is the midbrain. What we are going to do, if you notice the green color, there's several sections that have the green color. The midbrain, along with two parts of the hindbrain, are going to be lumped together into something called the brain stem. The mesencephalon we'll talk about very briefly because it doesn't have a huge function in humans. It's more like a connection station. Then in the hindbrain, those areas of the metencephalon and myelencephalon, this metencephalon is going to further divide into two very, very different regions. One is called the pons and one is called the cerebellum. We will spend a good amount of time talking about the cerebellum. The cerebellum is going to be very important in movement and how we do coordinated movement. The other two parts are going to be the pons and the medulla oblongata. Right? And we will take the midbrain, the pons, and the medulla, which will be continuous structures, and we can call them all the brainstem. Now please remember, we're only looking at the anterior most portion of the neural tube. The rest of the neural tube becomes the spinal cord. So the brain and the spinal cord are not two separate structures that are connected. They are one continuous structure. Okay? The spinal cord develops much less differentiation, but we'll see there is some differentiation in the spinal cord, right? And that will be later on in today's lecture. So for this portion, lots of new terminology. Okay, lots of new terminology. So you want to make sure that you are going through and understanding all of these different terms. Now, in addition to this exterior portion where we have the tissue here will be nervous tissue, so it will be mostly neurons and glial cells. We also have these spaces in between, and these are the true vesicles. Okay. These are going to come from that inner portion of the neural tube. They will become fluid-filled spaces, and these are going to be extremely important in how the brain is supported. So if you look inside at the blue parts, these two blue parts are going to be called what's known as the lateral ventricles. So you can almost imagine these as ventricles 1 and 2, the first two ventricles. It doesn't matter which one is 1 and which one is 2. Right. The reason I say that is because then here in the diencephalon we have the third ventricle. Then in the mesencephalon, which is going to be a very, very small area, we're going to have a little passageway of the fluid that's going to be called the cerebral aqueduct. Okay, and an aqueduct is an area where water or liquid is moved. Then in the hindbrain, we're going to have the fourth ventricle. Here would be the fourth ventricle. And then finally running through the spinal cord will be the central canal. Those ventricles are going to be connected to each other and to fluid that surrounds the brain. So we're going to see fluid inside the brain, inside the spinal cord, and around it. And that's going to be very important to how the brain is maintained. Anyway, so in looking at this linear layout of the brain, that wouldn't work very well in the way we are formed. So what we will see is that there's a space restriction, and that space is limited by the size of our skull, right, our cranium. Also, because we stand upright as opposed to walking on all fours, we're going to see that we have a slightly different orientation in our brain than a four-legged animal, say a sheep. So one of the things that's going to happen in terms of development, going from where we first see these distinct secondary brain vesicles, right, at about 5 weeks, to what it looks like at birth. Now, the human brain at birth looks like what the human brain looks like in an adult. It's just a little bit smaller. Okay. The human brain does not grow hugely compared to the body, right? If you think about how a baby's head is by the time they're 2, okay, compared to the way the rest of their body grows, their brain really doesn't grow that much, right? Their skull doesn't grow a whole heck of a lot. So as we go from having this linear brain pathway with our telencephalon, diencephalon, the mes, met, and myelencephalon, okay, this area here of the telencephalon, this is going to become the cerebrum. This is going to be the biggest part of the brain. This is going to grow the most. So in order for it to fit in with everything else, it's going to grow up and around, and it's going to actually encircle most of this. So at about 13 weeks, we see that the cerebrum has started to grow up, and it's surrounding the diencephalon. By about 26 weeks is fairly early on. Okay, you can see that the brain has, the cerebrum has folded. It's created a fold here. That's going to be very important in the adult brain structures. We start to see this creation of convolutions where the brain actually, to fit in and still have a lot of surface area, it makes little folds, and we'll talk about those. Okay, it's folding now completely over the diencephalon and part of the midbrain, or the mesencephalon, until finally by birth we have all of these complex convolutions. The cerebrum has grown up and around so that it fits into the human skull. The other part of the brain that grows quite a bit is this over here, which will become the cerebellum. The cerebellum grows up and out, and it kind of sits behind and fills the back part of the space below the cerebrum. Now, the diencephalon does not grow very much in size. It does develop and changes into the epithalamus, thalamus, hypothalamus, and a portion becomes the retina, but it doesn't get really big in size. Okay. The midbrain, the pons, the medulla, those don't get very large in size either. Okay. But what they do is, because of the way the folding is, they get kind of crammed together. That's why this region here, all shown in green, which is really the midbrain, the pons, and the medulla oblongata, all kind of get lumped into that category of the brainstem. So what this allows, it allows us to take a linear brain, fold it up and around. Now, the other big important fold here is, if you notice, our spinal cord runs at pretty much a 90-degree angle to the orientation of our cerebrum. Okay. This is going to be very different than if we look at a sheep, and we will look at sheep brains in lab. If you look at a sheep brain, their brain if you look at a sheep brain, the sheep brain's cerebrum and spinal cord are in the same plane, right? Because you can imagine the way their skull is, their skull comes out, and then their spinal cord comes straight out the back. Okay, so they have a much more linear plan. Anything that is a quadruped, four-footed, is like that. Now, in addition to this folding leading to differences in the external brain structures, remember we have those spaces inside, and those spaces are also going to be folded as well. So when the cerebrum folds up and around, the hollow tube inside of it, the ventricles, are also going to go up and around. So those lateral ventricles are going to have almost a giant C shape, and they're going to trace that development. So if you look, now you can see why these are called the lateral ventricles, because they kind of go out into the side. So here would be a lateral ventricle, and a lateral ventricle from an anterior view if you're looking face forward. If you look at a lateral view, you see that they come up and around. Okay, that is the lateral ventricles. The third ventricle, this would be located within the diencephalon, okay, and it's going to trace the shape of the diencephalon. The diencephalon is going to have an egg shape. It's going to look like paired egg structures. So you're going to see that the third ventricle has an egg-like shape. The midbrain is going to be very narrow and kind of compressed, and so it has a very narrow compressed ventricle. We actually call this cerebral aqueduct. And then finally, the brain stem here is kind of flattened out. So in the pons and the medulla, we have the fourth ventricle, and that fourth ventricle has kind of a flattened out shape. Now when we look at ventricles, you're just going to see the ventricles in the brain as a space, but they are actually full of fluid. The reason we can't see them full of fluid in a brain is because when you cut the brain open, the fluid leaks out. Okay? So we're just going to see a space, but that would be full of a fluid in a living organism, and it's actually an extremely important fluid. So the majority of the lecture that we're going to do about the brain is going to be on the cerebral hemispheres. And when most people talk about the brain, they're talking about the cerebrum. It's the biggest part of the brain. It makes up something like 83% of the mass. When it folds up and around, it creates those convolutions. Okay. We give those names. So the ridges, right, the parts that fold out, right, the mountains, okay, those are singularly called a gyrus, plural gyri. So we're going to actually go through and name some of those. We're going to talk about important gyri. Okay. And then we have those narrow grooves, those little valleys. Those are singularly called a sulcus, and plural are called sulci. There's also going to be very deep grooves, and the very deep grooves are going to be where the two hemispheres or two halves come together, and there's going to be one that runs between the two hemispheres, and that's called the longitudinal fissure. Okay? A fissure is basically a deep groove in something. There's also going to be a fissure if you imagine where the cerebrum comes up and folds. There's going to be a fissure there as well, and it's a deeper groove. Now in the cerebrum, there's going to be three main regions. We're really going to focus on the first two. Okay? The cortex is going to be gray matter. In chapter 11, we had talked about what gray matter was. Does anybody want to remind me what gray matter is? Robbie?
It's found in the spinal cord.
It's found in the spinal cord, but what is it? What makes it gray matter? In terms of the neurons. Let's compare it to the fact that I'm going to say we have cortex, which is gray matter, and then we have a separate part of the cerebrum that's called white matter. What's different about those two? The gray matter? Yes. So it's where we don't have myelin. So what that means, we either have entire neurons that are unmyelinated, or what else do we have? If it's not an entire neuron that's unmyelinated, what else would it be, the gray matter? Think about what part of a myelinated neuron could it be? Yeah. So the cortex is going to be gray matter. Gray matter is going to be where we have cell bodies. Okay. If we have cell bodies there, we also are going to have axon terminals. So the gray matter is where the connections are going to actually be. Okay. It's also where we'll have really tiny, short little association or interneurons will all be in the gray matter, because those little tiny neurons don't need myelin. So gray matter is where we don't see any myelin, which means that white matter is Myelin, right? The white matter is going to be where we see myelinated axons. So the white matter is going to be where we have, rather than the connections occurring, we're going to have the conductions occurring. So the white matter is really important because that's how the signals are passing. But you don't see a lot actually going on in the white matter, right? If you want to, look at the information that I have going on my computer, you're either going to come here and look at my computer screen, or you're going to look up at the projector, right? That would be analogous to the gray matter. It's where you can see stuff going on. It's the cell bodies. You are not going to come up and be like, what does she have going on? And stare at the cable, right? The information passes through the cable, but we can't see it. We can't really do anything with it, right? So the white matter is important because obviously if we cut this cable, we now are cutting off the information. So the white matter is important, but when we talk about function, we're really going to look at the gray matter. So we're really going to look at the cortex for the function. Okay. The other thing are what are called basal nuclei, and we'll only touch on the basal nuclei. Basal nuclei are regions of gray matter that are inside the cerebrum, right? The cortex is on the outside. That's what cortex means, okay? It's the layer on the outside. There are regions interior in the cerebrum that are going to be gray matter as well, and those are going to be other points of connection where we have cell bodies and axon terminals and little tiny interneurons. Okay? Alright, so we're going to start looking at the anatomy of the cerebrum. The first thing is there are going to be fairly deep sulci that are going to divide the hemispheres into what we call 5 lobes. Now, if you remember the bones of the cranium, these 5 lobes are going to be easy because we have the frontal lobe, which sits right below the frontal bone. Okay? We have the temporal lobes, which sit right below the temporal bone. We have the parietal lobe, which is right below the parietal bone. And we have the occipital bone that's right above the occipital lobe. Okay? So that's 4 of them. Right? There's also a 5th lobe called the insula. Right? And the insula is inside. Where there is this deep fold, if you could imagine the cerebrum coming up and around, it almost looks like if you were to fold your fingers over your thumb. Okay? There is a fold of cerebral cortex that gets folded inside then. And that's where the insula will be. So the other 4 are really mapped by the names of those bones. When we are talking about the brain is pretty unique in the sense of its anatomy in that you can't look at the brain and know what's going on in the brain. If I were to give you a box of bones and you've never seen bones again, you could figure out that these bones come together, and you could probably even put some of the bones together based on the way they're shaped, right? You could figure out what they do based on what they look like. Okay? With the brain, it's really hard because if you look at the part of the brain that controls speech, and the part of the brain that controls hearing, and the part of the brain that controls mathematical ability, they don't look any different. They're neurons. Okay? So in order for us to work our way around the anatomy and actually look at the function of the cerebrum, we have to build a map. Okay? We have to build basically a functional map, and we have to use landmarks to do it. So we're going to use anatomical regions to differentiate where functional areas are. So you're going to have to know both the anatomical term for a particular region of the brain and also what function goes there. So what I like to think of when I'm doing this is I always try, if you're trying to find your way around a new town, best way to find your way around a new town is find your way to Main Street, right? If you can find your way to Main Street, you could probably find your way all around the town from that point. So in the brain, Main Street is called the central sulcus. The central sulcus is going to be a kind of deep sulcus, pretty obvious, that's going to separate the frontal and parietal lobes. So when we're looking at this, we need to to be aware of where Main Street is. So all of the directions that I'm going to give you in terms of the function are going to kind of come from there. In terms of what we will be talking about with the function, this is all going to be housed in the cerebral cortex, right, in the gray. So we're looking at the outermost layer of the brain, right? We're going to talk about things like sensation, communication, memory, understanding, voluntary movement. All of these different functions are housed in the cerebral cortex. Now, one of the crazy things with the cerebrum, the cerebral cortex, is the hemispheres, right? Hemisphere means the half. Right? Each hemisphere acts contralaterally, so it controls the opposite side of the body. So when we're looking at the right hemisphere, it's actually going to be controlling the left side of the body and receiving impulses from the left side of the body. We'll see there's a crossover in the brainstem. The other interesting thing is that the hemispheres are not exactly equal. So for many things, we'll see that the right and the left hemisphere each has a motor portion, each has a somatosensory portion. But there are certain things that are on just one side of the brain, right? And we'll look at those. Now, as I said, we're talking about this in a kind of simplified manner. So I'm going to go through and I'm going to show you an area. I'm going to say this is the motor cortex. Okay? No part of the brain can act alone. Okay? The entirety of what we see in terms of conscious behavior involves many, many parts functioning simultaneously. Okay? And we won't be able to look at them all, so we're going to have to look at these things kind of simplified. Right? Look at a much simpler circuit to understand how things occur. And in terms of those functions, we're going to look at three main kinds of functions. The first two are kind of easy to understand. Okay. The motor area, I think, is the easiest for us to understand. This is where we have our control of voluntary movement. So here we're going to look at neurons we call motor neurons, and those motor neurons are going to control voluntary movement, so skeletal muscles. So we've already looked at this. We've seen this in Chapter 9. Now we're going to look at the previous stuff, right? We talked about neuromuscular junction. Now we're going to backtrack, and we're going to say, where does that come from? Right? How does that signaling occur? We'll also talk about sensory areas. And sensory areas are those areas of the cerebral cortex that allow us to become consciously aware of sensation. Okay? Now we have to kind of clarify sensation here. Sensation is a hugely variable term. There's a lot of different things that can be considered sensory. Okay? It's not necessarily good or bad. Okay? So we will mention both the general, what's called somatosensory, which is the body sensation. Think of this as the sense of touch. We will also talk about the special senses, so we'll see there is a region dedicated to vision, one to hearing, one to taste, one to smell. Now those, in a simple sense, when we looked at the nervous system, we said we have sensory information coming in and then motor response going out, and those are connected by association, right, by integration. Well, the association areas, that's the most complex of it all, because that's where we're going to have 5, 6, 7, 8 different parts of the brain all integrating information, right? The simplest thing, like putting an object in your hand and feeling it and then saying what that object is, you're using multiple parts of the brain. Okay? So the association areas are going to be integrating that diverse information, creating a response, processing tons of information that come in at the same time. So here we have a map of the cerebral cortex. The brain, if we were to take a hacksaw and chop off the top of somebody's head and take a look at the brain is not color-coded. The brain does not have an appearance that tells you what is one functional area versus what is another functional area versus another functional area. Which leads to a really interesting question. Right? How did we learn this? How do we learn what different regions of the brain do? All right, so in terms of looking at the functional areas of the cerebral cortex, here is our human brain. It is not color-coded in real life. You can ignore these numbers on it. We're not going to talk about what the numbers are. As we go through and look at this, we are going to look at these different functional regions. Right? There are going to be certain important anatomical regions that you're going to have to know, so you will have to know both the anatomical and the functional region. As I said, in terms of finding your way around, the best thing is to have a set point. I always use the central sulcus as my main street as my set point. Okay? So you see the central sulcus runs right here. The central sulcus is going to be the point of division between the frontal and parietal lobes. Okay? It's also going to separate these two very important gyri. We will call this the pre-central gyrus and the post-central gyrus, and that is relative to the central sulcus. If you can find these two gyri and this central sulcus, you can actually find your way around the brain pretty well. The brain, if we were to separate the cerebrum, right, we have the two cerebral hemispheres, so not only do we have this exterior part of the cerebral cortex, but part of the cortex also folds in into that longitudinal fissure. Okay? So we will also have regions within. Now this whole region here would be, you can imagine, would be inside the fold of the longitudinal fissure. This region that's shown as this kind of off-white tanny color, this will be part of the white, this area here called the corpus callosum. This is going to be a very important structure in our white matter. This is cut through. The corpus callosum is going to be a structure that connects the left and right halves or hemispheres. Okay? So in order to look at this view, we've cut through that corpus callosum. However, we have not cut through, we've just separated the two halves of the cerebrum. Now this is all labeled and color-coded, and what we will do is we go through and we talk about what the different functional areas do. You should come and flip back to this, and we will flip back to it. And in lab, you will then see a brain model. So the first thing we're going to talk about are the motor areas. Okay. The main motor areas we will talk about, there are four. We will look at the primary or somatic motor area, or motor cortex. We will look at the premotor cortex, and the premotor cortex is really the association area for motor. Right? We're also then going to look at two highly specialized motor areas. One is called Broca's area, and one is called the frontal eye field. So the primary motor cortex is located in the precentral gyrus. So this is the gyrus that is directly anterior or in front of the central sulcus. Okay. Here we have pyramidal cells. Right. Pyramidal cells, if you remember, we looked at those in lab in chapter 11. Those are multipolar cells. The axons that originate in the primary motor cortex will make up something called the corticospinal tracts. That will be part of the spinal cord. And if you look at corticospinal, that's going to connect the cortex to the spinal cord. So these will be the neurons that are going to connect the cerebral cortex to the spinal cord, which can then connect to the voluntary muscles. Okay. The primary motor cortex is going to allow the conscious control of voluntary movements, specifically precise and skilled voluntary movements. This shows us where we would find the primary motor cortex on the brain. So we're looking at the precentral gyrus, okay, which is that area that is just anterior or in front of the central sulcus. Okay. On your diagram it showed as red. What you can imagine is if we were to pull that cerebral cortex out, we could take a look at it, right? And due to our functional mapping, not only would we see that this whole gyrus here is going to be important in terms of our primary motor control, but we can actually specifically look at it in terms of what part is going to control what body part. Right? Now this is pretty cool. This is called a homunculus. You may have seen homunculus in different ways if you have, again, taken like a psychology course. What you can imagine here is if we were to take a section or a slice right out of that precentral gyrus and look at it and then draw on it what part of the human body is controlled by that area of the gyrus to scale, this is what we would see. Okay. So this region here, okay, this entirety, all of this central portion here, okay, all of the neurons that would be in here would be to control movements of the face. The brow, the eye, the face, the lips, the jaw, the tongue. Okay, that's a good full almost quarter to a third of this are just involved in movements here. Okay. If you look at the thumb, fingers, hand, a fairly good portion is dedicated to the thumb, fingers, and hand. Compare that to the area dedicated to the shoulder, hip, and trunk. Okay, so the shoulder, hip, and trunk, right, which is pretty much a good portion of the physical space of the body, has very little in terms of the amount of neurons dedicated to its movement. Well, think about why that is. How much can you actually move your trunk and hips? That's about it. Right? You can do a little wiggle. That's about it. Compare that to what you can do with your hands. We have much more motor control, much more movement in our hands. Same thing with our face, our mouth, for chewing, for speaking, for making funny faces. Okay. So if you have more surface area dedicated to a certain area, that means you have more neurons dedicated to a certain area, which means you have more control. Does that make sense? I will not require you to memorize this homunculus. Simply understand it. Another way you may see a homunculus drawn, which isn't as good, I don't think, a homunculus in terms of is sometimes drawn as an individual, and the body parts are drawn to scale based upon how much of the brain, how much of the cerebrum is dedicated to them. So this human, if we drew a human to scale, we'd have giant hands, a giant head, pretty small body, okay, for the motor homunculus. Now, no brain region can possibly operate alone. Anything that we say is a primary region, so the primary motor cortex, it has to have an association area backing it up. So the association area for this part is called the premotor cortex, but think of it as an association area. Okay. It's located just in anterior, so just in front of the precentral gyrus. This is where we have control of our learned, repetitious, or patterned motor skills. This is going to coordinate simultaneous or sequential actions. This is going to be involved in the planning of movements, and we're going to see that this part, this premotor cortex's association area, is highly dependent on information it's going to get from the cerebellum, which we'll talk about later. Is going to be involved in balance and things like that. So the premotor cortex is what's going to allow you to tap your head and rub your belly. If you can imagine what's going on as I pat my head and rub my belly, I have the different hemispheres of my brains doing two completely different motor skills, right? I can do it very well if I'm patting my head with my right hand and rubbing my belly with my left hand. If I switch it, it's much harder for me. I have to concentrate on it much more. This would also be where you have learned skills, repetitious skills, pattern motor skills, right? Anybody here drive a standard vehicle? Okay. When you first learned to drive a standard vehicle, what was it like? Yeah, it's like, okay, gas, brake, clutch, shift, gas, shift. Now you probably don't even think about it at all. Now you get into an automatic car and you accidentally go to hit the clutch. And you're like, where's the clutch? Okay, that's a patterned behavior, right? We have lots of pattern behavior, lots of things you don't have to think about in order to do. So that's also how we plan movements. You are not aware of the amount of information that gets processed every time you take a step. Right? If your brain wasn't processing a slew of information, every time you took a step, you'd fall down. Right? Because taking a step, you don't just lift your foot and put it down to take a step. You have to shift your weight, change how you're contracting your supporting leg, bring your leg forward, shift your weight again to put your leg down. All of those things happen using this part, the premotor cortex, as well as information that's going to be brought in from the cerebellum. Right? So those are two of the four motor areas that we'll talk about. The other two are going to be very specific motor areas. One is called Broca's area. Broca's area is located in the anterior region. It's just inferior, just below the premotor area. This is one of the areas of the brain that's usually only found on one of the two cerebral hemispheres. So usually in most people this is found on the left hemisphere. This is part of our speech area. This is a motor area that directs the muscles of the tongue. This is actually what anticipates what we are about to say and readies the muscles to say it. If you've ever heard of somebody who gets like tongue-tied, right, where they're talking so fast, and this happens to me all the time, where I start talking so fast, I start thinking so fast that I trip up on my words, right? So Broca's area is a motor control path. If you imagine trying to learn a new sound, right? Let's say we're all going to learn French and we have to learn how to make the rouge sound. Does anybody speak French? Can anybody do that rouge sound, right? Learning how to do that is pretty funny, right? Because we have learned how to use our tongues to make sounds that we are familiar with. When you have to learn how to use your tongue differently to make a new sound, it's actually quite complex. Trying to teach somebody to roll their R's in Spanish, like ferrocarril. How many of you can roll your R's? Can anybody do that? Right? And some of you are like, you know. Think about trying to teach a little kid to talk. Okay, little kids who are first learning to say some of the harder sounds like F and S. Right? Think about somebody whose native language isn't isn't English, trying to say the TH sound. The. We can all say the. Do you know how hard it is for people to say the? That is why most people who don't speak English say the. Right? They don't say the, because it's a very hard thing. So that's all coming from Broca's area. Those are very, very fine movements of your tongue. There is something called Broca's aphasia, and what this is the inability to speak due to the inability to correctly make the muscle movements to produce speech. The last one is going to be located just anterior to the premotor cortex and above Broca's area, and that's going to control voluntary eye movement. We call this the frontal eye field. Now, eye movement is very complex because we have both voluntary eye movement. This is eye movement when you're looking around and moving your eyes and rolling your eyes. But we also have tons of involuntary movement of our eyes as well that we do not control. So eyes really have kind of two layers of control to them. The frontal eye field is what's going to control the voluntary movement. So flipping back just to get your bearings again, here is the central sulcus. This is the pre-central gyrus. This is the primary motor area. Just in front of it we have the the premotor cortex, which is the area that is the association area. Just in front of that we have the frontal eye field. Okay. Below that we have Broca's area. Remember, Broca's area is usually only on one hemisphere, usually the left. Okay, so if we take a look in the posterior direction from that central sulcus, we're going to look at some sensory areas. The four main sensory areas we are going to look at is the primary somatosensory cortex, the somatosensory association cortex. Okay, we'll look at briefly primary visual and visual association areas, as well as primary auditory and auditory association areas with a special area in there. And we'll just quickly point out the olfactory, gustatory, and vestibular cortices. The plural of cortex is cortices, okay, not cortexes. So once again, get our bearings. Here is the central sulcus. We are now going to the post-central gyrus, that is the anatomical term, and that is where we find the functional region known as the primary somatosensory cortex. Somatic sensation or somatosensation, this has to do with body sensation. So you can loosely translate this to the sense of touch. So this is where sensation coming from the body is going to come in and be perceived. This is going to receive information from the skin, skeletal muscles. Here's where we'll see something called spatial discrimination. Okay. Spatial discrimination is the ability to tell two separate points. Right? We have pretty varied spatial discrimination. We have very good spatial discrimination on our hands and very poor spatial discrimination in other parts of the body. Right? You could imagine if I gave you an object and I said, feel this object and tell me what it is. You're going to put it in your hand and feel it. Right? You're not going to like rub it on your belly to feel what it is. Okay? Part of that, if we take a look at the somatosensory cortex homunculus, set up the same way, taking a section out, now what we see is the amount of that region of the postcentral gyrus that's dedicated to somatosensory in a certain body part. Okay. Once again, we have a very large amount that's dedicated to the face, the nose, the lips, the eye. We also have a lot dedicated to the teeth, the jaw, a little bit to the gums. Okay. The hands, right? The hands, the fingers and the thumb have a fairly large region compared to, say, the trunk. The entirety of the trunk has less somatosensory input than, say, the fingers and the thumb. Okay. So we have much better feeling, much better spatial discrimination in the hands than in the trunk. Right? Now think about babies. If a baby wants to explore an object, where does it go? In its mouth. Okay. We actually have quite a bit of sensation in our mouth. Babies are not actually trying to taste things when they put it in their mouth. That's how they explore things. Okay. That lets them know, is it hard? Is it soft? Right? Of course there's taste, but most of it isn't about taste, right? Because if you put something in your mouth that doesn't taste good, you take it out of your mouth. Babies will keep it in their mouth, right? It feels hard, it feels bumpy, it feels lumpy. It's a really cool sensation for them. So this would be where we would have our primary somatosensory homunculus, and you can look at the relative areas that are dedicated to the different sensations. Okay. Now the somatosensory association cortex, this is going to be very important. You can imagine that remember, we have sensation coming in. So the primary somatosensory cortex is receiving information. It's receiving tons of information, and it's receiving raw information. So that information has to be processed. It has to be integrated. That's going to occur in the somatosensory association cortex. This is going to be located just posterior to the primary somatosensory cortex. It is going to be where we have integration of sensory information. This is where we have the first step in understanding the sensation, okay? In terms of the size of thing, the texture of thing, the relationships of things, right? This is going to be the first step in understanding what something is. You can imagine if I told you to close your eyes and I put an object in your hand, the raw information that's going to go into the somatosensory cortex is just bits of information. It's hard, it's cold, it's square, right? The processing for you to actually figure out what it is that you're holding in your hand involves tons of different parts. You have to be able to imagine what this thing looks like in 3 dimensions. You have to give it a name. You have to be able to understand the relationship between the parts. So those are going to be 2 of the big association cortices. Okay. Then we have the visual area. The visual area is also called the striate cortex. This is seen on the very posterior tip of the occipital lobe. The majority of it is buried in a deep sulcus called the calcarine sulcus. This is where information from the retina comes in. You do not see with your eyes. You see with your brain. You see with your cerebrum. You see with your primary visual cortex. The retina of the eyes simply picks up photons of light. The photons of light activate neurons. Those neurons send information down axons. Those axons are then received in the primary visual cortex. That's where the first step of processing those images gets put together. Okay. When that comes in to the primary visual cortex, it then has to go, the information then has to go to the visual association area to be processed, to interpret the stimuli in terms of what is color, form, shape, movement. Okay? Then, of course, there's many other brain regions in order to put pictures together and to tell stories and put names on things. Okay? So that would be the visual area. Now, the visual area, if we take a look at the map, here is the primary visual cortex. This is the visual association area. If we look at the separated hemispheres, we see the primary visual cortex actually goes up very deep into this calcarean sulcus. Okay. You can almost imagine a pathway, right? The eye would sit approximately here, and there's going to be a pathway with those optic nerves not shown in this diagram. The optic nerve is going to come in, it's going to cross at that optic chiasm, which we've seen before, and it's going to go back, it's going to pass through an area here called the thalamus, which we'll talk about, and it's going to end up here. Now, if you've ever seen a cartoon where the character falls down and hits its head and starts seeing stars or birdies, okay, that actually has some real basis. If you've ever taken a good wallop to the back of your head, you may actually see stars. Okay? What that is sometimes neurons can be induced to send an action potential from the wrong type of stimulus. So rather than responding to another neuron, if you whap the back of the head, some of those neurons in the visual cortex are going to start sending signals. So they're going to start telling your brain that it's seeing something. Okay, there's not really anything there. Okay, and that's how you get that. If you've ever hit the back of your head, you actually can see stars. Okay, there are cases where people have fallen and taken a severe blow to the back of the head and lose their vision. Right, they go blind. There was a movie made about it 5-10 years ago. It was called like Ice Princesses or something silly like that. And it's about a girl who ice skates and she falls down, bangs her head, becomes blind, continues ice skating, falls in love. Yay! So so it is something that can happen. So that would be the visual areas, the primary and visual association areas. Now the auditory areas, this is where we have our sense of hearing. Okay? The primary auditory cortex, this is going to be located in the superior margin of the temporal lobe. Okay, so if you remember where the temporal bone is, the temporal bone has the external acoustic meatus, which is going to lead into the cranium, is going to lead to the parts of the brain that are going to receive our information. The primary auditory cortex is going to receive very basic information about pitch, rhythm, and loudness. It's the association area that is going to process sound. So what is that sound, right? A bang like that, right? Your brain, if you didn't have your eyes open and you heard me banging, you would know that it was somebody banging on a door or a wall. Okay? The actual perception of sound is going to come from the association area. Now, there's a very specific part of the auditory association area called Wernicke's area. And we will talk a little bit about Wernicke's area when we talk about the general association areas. But Wernicke's area is part of the brain that is involved in the understanding of speech and comprehension of speech, sounding out of words, understanding of words. There is a difference in the way we perceive sound versus the way we perceive words. Okay, and that usually comes from Wernicke's area. And Wernicke's area is actually also one of those cerebral cortex regions that is found only on one hemisphere. The same hemisphere usually is Broca's area, so in most people, the left hemisphere. Now, there are other areas. If we take a look, the primary auditory cortex is located here. If you can imagine superimposing the skull and then the soft tissue structures, you would see that the ear would be right about there. Okay, this would be then Wernicke's area. It's a more generalized area. The other sensory areas If we look at the other sensory areas, we won't talk about them too much. They are more folded in, more hidden. Okay. The primary olfactory cortex is located in this area here that's called the uncus. Okay. The sensation of smell, olfaction, is one of the most primitive of the senses. It's one of the most basal of the senses. It's also one of the senses that's most closely located with other regions or other functions of the cerebrum, things like memory storage. And that's mostly because of the way it's located and how it's located. It's going to be tightly interacted with something called the limbic system, which we will talk about. Okay. So that's where you're going to see the main primary olfactory cortex. In addition to the motor, what would be the output side of things, the sensory, which would be the input side of things, we also have a lot of association areas. Okay. We can break some of these association areas down. They get more and more complex. This is harder to understand because there's no set rules per se. This is how we connect all of these other things. We have to have connections here. If we don't have connections, signals stop, and then they are lost. Okay. The prefrontal cortex, okay, which is this area down here, you could almost imagine this being right above your eyes. Right? If you were to stick your finger in and go up. The prefrontal cortex, this is where personality is believed to be housed. That's where the individual mentioned in your book, Phineas Gage, he had damage to his prefrontal cortex. There is some research that suggests certain people with severe antisocial, psychotic, sociopathic behavior may have damage to the prefrontal cortex, and that damage to the prefrontal cortex is what leads to them having these severe personality disorders. Okay. In this area here, we also have things like problem solving, working memory for object recall. So what happened, what did you see just a few seconds ago, right? Then we also have these kind of diverse regions that are harder to label, right? How you think about things, right? The idea of your conscious mind thinking about things, how are you? Most of that is probably seated in that area of the prefrontal cortex. Okay. This is where you have things like judgment, reasoning, intellect, cognition, recall. These are all going to be very closely linked to the limbic system. The limbic system is the emotional part of the brain. We will talk about it very briefly. It is, of the parts of the brain that are not terribly well understood, I'd say the limbic system is probably one that is the least well understood because it is the hardest to understand, especially in humans, because emotion is so complex. Now, language areas, we've mentioned two of them already, Wernicke's area and Broca's area. Wernicke's area is sensory, that's for sounding out unfamiliar words. Broca's area is where we have speech preparation and production. Okay. We also have, in the lateral prefrontal cortex, we have language comprehension and word analysis. This is not just sounding out unfamiliar words. This is understanding when words come in that they have meaning. What does that word mean? Not just meaning, but it's an association area. So if I say the word elephant, okay, you hear the word elephant. That word elephant then you sound out, then you put meaning on it. Now that is then going to set off a whole chain of events. If I say to you, don't imagine an elephant right now, I bet all of you just had an image of an elephant that flashed through your mind. Okay, that's because your hearing centers are inextricably linked to your perception centers, your visual centers. Okay, so that's a more complex thing. So we see a coordination between auditory and visual aspects of language. Okay, in terms of if you hear a word, you can visualize what that object is. If I show you an object, you can verbalize what that object is. Right? These are things that can go wrong. I mentioned before, Oliver Sacks writes a series of books, and this is one of the things that he looks at is when that system goes wrong. where I show you a picture of an elephant, you say peaches. Okay, and you know what that object is, but the words get mixed up. Now that leads us into something important in terms of that lateralization of cortical function. What I mean by that is the hemispheres of the cerebrum don't necessarily do the same job the same way. Okay. Each hemisphere has certain abilities that's not shared with the other hemisphere. We talk about cerebral dominance. Cerebral dominance is when the hemisphere that you have that is dominant for language is your dominant hemisphere. Okay. Now, overall, if you think about the division of labor between your hemispheres, okay, the left hemisphere of the brain tends to control what you would think of as your more math, science, analytical, logical things. The right hemisphere, salud. The right hemisphere controls more visual spatial skills, emotion, and artistic skills. This is where the concept of people being right-brained or left-brained comes from. Everyone does have brain dominance. Okay? Everyone has brain dominance, and what that means is that your speech centers are on one hemisphere or the other. Okay? If your speech hemispheres are on the left side, you may tend to be more left-brained in the sense of having better math skills, better language skills, better logic skills. If your language side is on the right brain, you may have better visual spatial skills, emotional skills, artistic skills. Okay? With very few exceptions, there are no people that are completely right-brained and completely left-brained. Okay? It's a dominance, right? You're better at it. Okay? Think about it with right and left-handedness. Now, right and left-handedness is actually not controlled by the same thing. Most of us are probably right-handed in this room. Most of us write with our right hand, do most things with the right hand. But if you broke your right arm, you would not be completely useless, right? It would be harder, but you could brush your teeth. You could hopefully wipe yourself. You could type. You might chicken type, but, you can do it. It's the same thing with our brain in the sense of you may be left brain dominant. You may have an easier time with science and math and logic, but you have some degree of emotion, intellect, things like that. Okay. So that's just an overview of what was previously mentioned in terms of the lateralization of cortical function. Okay. Let's pause here. Let's take like a 15-minute. In looking at the cerebral cortex, we're looking at gray area. So we have areas where we have cell bodies and we have the actual connections between cell bodies and the axon terminals of the previous neurons. And that's all well and good, and we look at the functional areas. But in order for those functional areas to have any sort of function, they have to be connected to other areas. And so those conducting areas or the communication areas, that's where we're going to see white matter being important. Okay. Within the central nervous system, we are going to talk about axons that are myelinated. We're going to call those tracts. So where we have sections that are axons that are all myelinated, we're going to call those tracts. That's different than what we'll see in the peripheral nervous system. We'll actually use a different term for that in the peripheral nervous system. This is going to allow for communication between different areas of the cerebral cortex and then from the cerebral cortex to lower brain areas, right? So from the cerebral cortex to things like the cerebellum and the brainstem, and then also to the spinal cord. Now, there are three main types of tracts that we are going to talk about, or three main types of fibers that we're going to talk about. One is called commissures. And when you have a commissure, commissures are going to connect gray area in the two different hemispheres. So they're going to connect left to right and right to left. Right. We're going to talk about one main one, and that is going to be the commissure that will connect the two halves. We will see that there are commissures in not just the brain, but also in the spinal cord, which is part of the central nervous system. Right. The main one we'll see in the brain is called the corpus callosum. we also then are going to have association fibers. And what association fibers are going to do, they are going to connect and associate different cerebral cortices within the same hemisphere. So this is where you would have the communication between the primary visual cortex and the visual association cortex, or between the visual association cortex and the auditory cortex. Okay, so these are going to be association fibers. They're going to associate two different areas. The third kind then are projection fibers, and these projection fibers are going to enter the hemisphere and also leave the hemisphere from what are considered lower brain areas or the spinal cord. And now when we talk about lower brain areas, you have to imagine it's not necessarily a spatial thing. It's more of a complexity thing, but it also happens to be spatial. So if you imagine the cerebrum being on top, below it then you have the diencephalon, below it then you have the brainstem, right? So when we talk about higher and lower brain areas, we mean higher in terms of complexity, but it actually winds up being higher spatially as well. So those projection fibers are going to help connect those. So if we take a look at a section of a brain, now this section of the brain is not a sagittal section, right? A mid-sagittal section would separate the brain directly in half, and we would not see white because we'd separate the cerebral hemispheres and we'd be seeing the cortex where the cortex folds over. So instead what we're doing is we're cutting the brain in a sagittal section, a slightly off sagittal section. We're actually going to cut through it so we can see the gray and the white. So here the gray looks kind of dark yellowish, and the white looks kind of whitish. Okay, they're not actually gray and white. It's not that easy. This shows us association fibers. So association fibers, association fibers would connect from one region to another region to another region, all within the same hemisphere. The projection fibers are going to connect from lower brain regions. They're going to pass through this structure here. They're going to go through something called the thalamus, and they're going to project up to the cerebral cortex. We're also going to see these that look like little green dots. These, you can almost imagine, are fibers coming into and out of the board. So when we cut them in half, all we see are the little cross sections. These would be part of the corpus callosum, and this would be the commissure that connects the right and left halves of the cerebrum, or the right and left hemispheres. Now we will look at, in lab today, we will look at a sheep brain, and we will see some cut regions of the sheep brain, and you will be able to differentiate between the white and the gray matter. Again, it's not exactly white and gray. This would be a human brain that is cut in that same fashion as the diagram we just looked at. Now I do have a human brain that we will look at as a display, but we will not be able to touch it or play with it or move it. Okay. Here, though, you can see the cerebral cortex all along the edge. Okay. The cerebral cortex has a darker appearance, so this is where we'd have our cell bodies. Okay, and this would be the white. And you can actually almost make out the fibers here. Okay. The association fibers would go from this point to this point. The commissure fibers would go across, and then these would be the projection fibers. So that white matter is really important. That concludes kind of the overview of the cerebrum. We're going to spend a whole bunch of time in lab today just looking at the brains. Any questions, though, before we move on to the lower, quote-unquote, brain regions? Yeah.
You said, like, if you cut the brain in half, like, right at the hemisphere, you would not see the white?
You'd only see the white if you cut. You wouldn't see the white within the cerebrum. You'd see the white of the corpus callosum. So that is a continuous part. So if you could imagine your cerebrum, and when you see the models, this will be much more clear. Your cerebral hemispheres are shaped like this, like a walnut, right, where you can separate the two halves. But the inner part that holds them together is the corpus callosum. That is continuous fibers that cross. So in order to separate it, you do have to cut through that. And when you look at the models, that you'll see that, and when we look at the sheep brain, you'll see that. Okay? Other questions? All right. So then the next region is the diencephalon. The diencephalon does come from that embryonic forebrain or prosencephalon. The diencephalon is going to be really made of three main paired structures. So we have one on each side. Okay. We have the thalamus, the hypothalamus, and the epithalamus. This is also where the retina will develop from, but we won't really talk about the retina at all. Enclosed within the diencephalon, we're going to see the third ventricle. If we think about what the thalamus is, the thalamus, we have these paired egg-shaped masses. These are going to be connected at the midline. They have within them 4 groups of nuclei. Now, in the central nervous system, when we use the term nucleus or nuclei in the central nervous system, we mean an area of gray tissue, right? An area of gray matter. So it means an internal area where you have lots of cell bodies, so therefore lots of the connections. Okay. So this is an area that's going to function the way we would see the cerebral cortex functioning, but it's internal. Okay, so it's within the structures. Now, I will not go through and I will not test you on the names of those different nuclei. Each one has a different name and it has a different function, and the naming is basically based on anatomy of where it's located. Okay? If you go through and you read the textbook, your textbook does go more in depth into those than I will be. What the thalamus does is it's going to receive fibers from the cerebral cortex, and it's going to project fibers into the cerebral cortex. This is going to basically act like a editing station or a relay station for sensory information that's coming in from the body. Has to pass through the thalamus first, and the thalamus helps to route that information and edit that information and relay that information to the cerebral cortex. Okay. So anything that comes in terms of being sensory has to pass through the thalamus before it gets to the cortex. Okay. This is going to be important then in most of our behaviors. Okay. It's going to mediate sensation, right, how information comes in, and it's also going to have a role then in things like motor activities and learning and memory, and we'll talk about how those are all related to sensation. Right. So that would be the thalamus. Just below the thalamus, then, we have the hypothalamus. Its name actually means below the thalamus. Okay. Here we also have a whole bunch of nuclei. The hypothalamus is roughly triangular in shape. This is where we are going to have most of the control of the viscera of the body. Okay, so how the body works. The hypothalamus has its little fingers in everything. Okay, the hypothalamus is the part of the brain that's going to talk to the pituitary gland. The pituitary gland is considered the master gland, and it really sets off almost everything in your endocrine system. So this is where we see a really tight interaction between the nervous system and the endocrine system, the fast and slow control systems of the body. Okay. Now, in terms of looking at this on the brain, there is a key landmark. They are called mammillary bodies. Okay. These are 2 small paired nuclei that bulge out just behind the point of the hypothalamus. Okay. Those are going to be involved in the olfactory pathways, but they're also a good landmark. If you look at mammillary bodies, what does the word mammillary make you think of? Mammary glands, right? Or breasts or boobies, right? And when you flip the brain over and look at it, you're going to see 2 little structures that look like tiny little Barbie doll boobies, right? Those are the mammillary bodies. Once you find those, that's going to help direct you as to where the rest of the diencephalon is, because that forms the back part, okay? So this would be the mammillary body right here. That's going to direct you to where the hypothalamus is. The hypothalamus is below the thalamus, so that would direct you to where the thalamus is. Okay. Now there is a structure that comes off of this. This is called the pituitary gland or the hypophysis. Okay. Pituitary gland is the more common usage. The pituitary gland is really 2 glands in one, and it releases a slew of different hormones that's going to affect most of your body chemistry and most of your homeostatic feedback mechanisms. Okay. So things like blood pressure and breathing and heart rate and digestive tract motility, body temperature, hunger, satiety, sleep, these all have some influence or are influenced by in some way the hypothalamus. The hypothalamus also is going to be part of something called the limbic system, and the limbic system involves emotion and memory. So you'll see that the hypothalamus is also going to have a role in things like emotions, pleasure, fear, rage, and memory as well. So the hypothalamus does a lot of stuff. We've actually mentioned the hypothalamus specifically before. Do you remember when we talked about positive feedback? We mentioned oxytocin being important in the positive feedback loop of childbirth. Okay, the hypothalamus is actually producing that oxytocin. That oxytocin gets released from the posterior pituitary. So here we see a really tight connection between the hypothalamus and that pituitary gland. Now, we are not going to go in depth into endocrine function, but this is really where we see an inextricable link between nervous system and endocrine system. Now, the third part of the diencephalon is the epithalamus. Okay, epithalamus meaning above the thalamus. This is really where we have two main parts of the epithalamus that are going to be important. Here we're going to have the pineal gland. The pineal gland is going to be responsible for the release of melatonin. Melatonin is a hormone. It's important in sleep-wake cycles. It's important in mood, sleep regulation. The other thing that I would say is even more important is here in the epithalamus is where we're going to find a choroid plexus. A choroid plexus is a structure that is going to secrete cerebral spinal fluid. Cerebral spinal fluid is that fluid I've been alluding to that fills the ventricles, fills the central canal of the spinal cord, and surrounds the brain and spinal cord. It's going to be very important in providing nutrition to the brain and spinal cord, taking away wastes, floating the brain. Okay. And one place where it's actually created or produced is in this region near the third ventricle cord called the choroid plexus. So now if we take a look at the human brain, just so we can get our bearings, you cannot see the diencephalon in an intact brain. The reason for this is the cerebrum has grown up and around it. Okay, so you have to just get your bearings. Right? This is if we are looking at the ventral aspect of the brain, so we're looking at the brain upside down. Okay. This would all be cerebral cortex here. Okay, so we have the cerebrum. Just to get your bearings, this would be where you have the spinal cord. These are the sections we'll talk about in a minute which make up the brain stem, which are actually the medulla oblongata, the pons, and the midbrain. This region here we'll talk about momentarily. This is the cerebellum. Okay. Here are the mammillary bodies. Okay. If you can find those mammillary bodies are showing you where the hypothalamus is. So the hypothalamus is here. So if you can imagine, where would the thalamus be? It's inside here. It goes inside, right? The cerebrum wraps around it. That's why you don't see it on one of these. Okay. We do have some models that show us what it would look like if we kind of peeled the cerebrum off. Another important landmark for you to take a look at is this is the optic nerve. Okay. We are, in Chapter 13, going to look at all the cranial nerves. The optic nerve is one of the cranial nerves, but it also is a very good landmark, right? This is where you'd have one optic nerve going to one eye, another optic nerve going to the other eye, and then they're going to cross in the middle, and that's called the optic chiasma. Then those tracts go back into the brain. So it almost looks like an X. It's a cross. That's another important landmark in terms of finding your way around the brain. So this is also a good way to come back and look at the brain. Now if we move down to the brain stem, here we have three areas that we kind of lump together. Okay, the midbrain, which comes from the embryonic mesencephalon. We also have the pons and the medulla oblongata, which come from the embryonic metencephalon and myelencephalon, which originated from the rhombencephalon. Here on this diagram, we have pulled the cerebrum off. This shows us the diencephalon. Okay. Here would be the pituitary gland. This would be the hypothalamus, the thalamus. And then we're going to see that this region that comes around, that's going to be something called the hippocampus, which we'll talk about in a bit. The brainstem then is shown in green. Right? So this would be the midbrain. This would be the pons. This would be the medulla. This is where we have control of our automatic behaviors which are necessary for survival, things like our breathing reflex. We can consciously increase the rate of breathing and slow the rate of breathing, but you cannot hold your breath so long that you actually die. Okay? You may hold your breath so long that you pass out, at which point your conscious control goes away and breathing will kick back in. I have a 2-year-old, so I have to constantly remind myself of this because my child can scream so long and then hold her breath so long that I think she's going to die. But I promise you she won't. This is the area that if there is damage to the brainstem, it usually results in death. Okay. This is where we have also the pathway for the tracts between the higher and lower brain centers. Okay. So if you can see these ridges are showing us basically the directions that the fiber travels. So it helps you to map out which way the connections are going. The other thing that's important here is if you see these little yellow projections coming off, these are called cranial nerves. And we'll talk about cranial nerves in chapter 13. Cranial nerves are nerves that come directly off the brain that are going to innervate part of the body, usually the head and face, and that's part of the peripheral nervous system. There are 12 pairs of cranial nerves, and 10 of those pairs come off of the brainstem. Okay. So if there's damage to the brainstem, that's going to lead to lots of issues in terms of how those cranial nerves function. Now the pons, we don't talk a lot about the midbrain. The midbrain really is just going to form a pathway. Now it's important. The midbrain is important. It's a very important pathway. It doesn't do a whole lot. In a way, it's kind of like a bridge. You don't really think about what a bridge does until the bridge isn't there anymore. Then you don't have a pathway. The pons then, okay, if you notice the pons has fibers that look like they run the opposite way, because what the pons is going to do, fibers are going to come in and they're going to run back to the cerebellum. The cerebellum sits right behind the pons. So the pons is going to be where you're going to see connections between the cerebellum, which we will talk about momentarily, and the cerebrum. The medulla oblongata, then, this is the most inferior portion of the brainstem. This is where the brain becomes the spinal cord. Here we have these two ridges we call pyramids. Okay, these pyramids, these are ridges that are formed by what are called corticospinal tracts. A tract is where we have axons. Corticospinal, these are axons that go from the cortex to the spinal cord. These are called pyramids, not because they are pyramidal in shape, but because they are made of the axons of pyramidal neurons. Okay. The pyramidal neurons, those pyramidal cells, we looked at those. Those are part of the cerebrum, the cerebral cortex. Now there is a crossover point shown by this jagged line here. This is the decussation of the pyramids. This is where we have the crossover of the corticospinal tracts. So this is the point where we see the left brain controlling right body crossing over. This is where the crossover occurs. So then from that point on, the spinal cord then controls the right side of the body if it's on the right side of the spinal cord. All right? Now the medulla oblongata is really important. If we don't have the medulla oblongata, if it is damaged, we lose communication with the spinal cord. The last part of the brain that I'm going to talk about is the cerebellum. It's actually the second largest portion of the brain by mass. It makes up about 11% of the mass of the brain. It's located just behind the pons and the medulla of the brainstem and just below the occipital lobes of the cerebrum. Okay, the cerebellum is going to sit in that space that's created by the occipital bone. The cerebellum is going to be very important, but it functions subconsciously, so you're not aware of what your cerebellum is doing. It allows for precise timing and appropriate patterns of skeletal muscle contraction. So skeletal muscles, the way we use them, you're not, in order to simply move your leg, you're not just contracting one muscle, right? You're contracting many muscles, you're coordinating them. You also have to, in order to flex your arm, okay, or you're contracting your bicep, you also have to relax your tricep. So those have to be timed appropriately. Okay. The cerebellum in humans is bilaterally symmetrical, right? So we have two hemispheres. We will see in some other organisms things that don't have good division of their left and right body control. They have a single cerebellum. So we look at a sheep brain. A sheep has a single cerebellum. These have a different folding. They have folding increasing surface area, but they have a different folding pattern. So they fold into what are called folia. Okay, so these are the gyri here are oriented more transversely, right? So horizontally, if you imagine a person standing up. There is also gray and white matter here. Right? The gray would be on the outside, the white is on the inside. If you cut through the cerebellum, you see what's something called the arbor vitae, which literally means a tree of life. And you can see that there almost looks like a tree-like pattern. So we see much more branching of the white matter than we see in the cerebrum. Okay, and that would be the cerebellum. Now what the cerebellum does is it's going to receive impulses about the voluntary intent to make a muscle contraction. So when you think about contracting your muscles, okay, when you're consciously thinking about moving, the cerebellum receives that information and basically says, okay, what do we need to do? What has to happen in the body for this movement to occur? And it takes in all of this information from things called proprioceptors, which sense your body position. It also uses visual signaling. It also uses vestibular signaling. Vestibular is your sense of balance. It takes all of this information and it actually calculates the best way to do that movement, right? So if you're going to take a step, it takes in all of that information. That's why if you're walking on a flat surface, if you look at the way your muscles contract, working on a flat surface versus walking upstairs, you don't necessarily have to think about the differences, but there are differences there. If you have ever been walking upstairs and you think there is one more step than there really is, and you go to step up a step that's not there, you fall over, right? That's because for some reason you've tricked your cerebellum. You think there's one more step. Your cerebellum is preparing to go up one more step. It's not there. Okay. So what the cerebellum does is it creates this blueprint of this is the way we should do this movement, sends it over then to the premotor cortex and then to the primary motor cortex to actually do the movement. Now, of course, this happens so quickly that you don't become consciously aware of it. But when you have an individual who's learning to walk, a child, if you are going to do physical therapy and you have somebody who has an injury and they're learning to walk, this is a process for them, right? Physically, they have to really, their muscle movement is slow. They have to think about what they're doing so they don't fall over. Okay. What happens with us, we've been walking for a long period of time, those blueprints are all pretty much ready to go, right? They're all kind of chalked in there and ready to go.